My name is Alphonse. I'm a first year PhD student. I'm going to talk about my work, which consists in the identification and characterization of unannotated human snoRNAs using an integrative transcriptomics strategy. snoRNAs are no-coding RNAs present in eukaryotes, known for their involvement in ribosome and ribosome-based and gene expression regulation. Generally, snoRNAs are located in intron of longer genes or intergenic region, although depending on the organisms. In humans, the majority of snoRNA are poorly characterized, or recent studies indicate that many annotated snoRNA are not explicit, while others are still from missing from annotation. So, in this project, we aim to carry out a much wider screen to test the completeness of human snoRNAs. Um, for the the method, here the method, we're working on TIGA-seq data from seven human tissue to first identify predicted annotated genes in the set database having a size between 15 to 200 nucleotide and having an entronic or intergenic localization. Then for these genes that have that we will call unannotated genes, we have evaluated their expression level and keep those that have a TPM greater than 1. Then we have identified among these genes those that bind the snoRNA binding proteins and those that have affected by the depletion of discarding. All of this data were integrated to identify potential box snoRNA or box HCC snoRNA. About the results, this first figure shows the number of predicted unannotated transcripts with a TPM greater than 1 in each sample. Particularly in this figure, this one, we see a strong representation of here of these two samples. In this other figure, this one, we have the number of predicted unannotated transcripts with a TPM greater than 1 in at least one sample, because some transcript can be found in several different samples. In the figure 2, here you can see the unannotated genes significantly underexpressed after a depletion of discarding compared to leucofactorin has control, and you can see on the 6,170 predicted transcript, 309 unannotated genes that are affected by this depletion. Then in the figure 3 here, you have the result of the integration of RNA binding protein immunoprecipitation sequences and depletion analysis data to identify potential snoRNA. For this figure, this first figure, we have the intersection of the result of NOP56, 58, and fibrillary paraclip for the identification of box snoRNAs. And for this other one, this figure, the identification of HCCA box by the intersection of discarding eclipse, discarding paraclip, depletion of this protein. The unannotated positive genes positive to depletion and to at least one immunoprecipitation test are selected to be explored as potential snoRNA that we can see target here in blue. Sorry. As you can see also, this NOP58 depletion data has not been integrated for the case of box snoRNAs, which is expected because this experiment is still progress. So without this data, we have so far identified 610 unannotated genes related to the RNA binding protein of box snoRNA, and for box HACA, we have identified 21 no unannotated genes that are affected by discarding depletion and that bind at least one RNA binding proteins. For these 21 unannotated genes, here in this figure, 66.7% are located in introns of unannotated genes, of which 17.8% are protein coding genes. Also, you can see in this figure that some of these genes are more expressed in specific tissue, has the case of this genes, which is strongly expressed in skeletal muscle. You can also see almost all of these unannotated genes that can be potential box HCC snoRNA have a low conservation level. Finally, just 21 unannotated genes have been tested on HCCA and box HCCA predictors snoRNA, namely snoReport and snoGPS. And however, we have here in this figure a low prediction of which 6.62 per 21 for snoGPS, and just only unannotated genes predicted to be a box HCC snoRNA by snoReport. To conclude, this approach allowed us to identify 21 non-annotated genes which are explicit in at least one of the tissues, and so are affected by the depletion of biscarin and bind also to these proteins. These 21 genes are proposed to has a box CD, box HCC snoRNA to be explored and characterized. Then 610 no annotated genes are bound by box CD snoRNA binding protein, and integration of the NOP58 depletion data would allow to identify the most likely CD snoRNAs missing from annotation. This result further demonstrates the interest of implementing more efficient and more reliable pipeline for the identification of snoRNAs. Thank you.